Okay, let's go ahead and go over the urinary system a little bit. Hopefully you have your notes again, so get your notes out. First things first, the urinary system. The primary function is to keep the body in homeostasis by controlling the composition and volume of the blood. The lungs, skin, and digestive tract also function in waste elimination. The lungs from breathing out carbon dioxide and metabolic water. Some of that's metabolic water. The skin from sweat, you get rid of water and some waste materials, and even salts. And the digestive tract, undigestible materials are gotten rid of by way of a form called feces. The system contains two kidneys. You can see right here, let me get a little pointer out. You have a right kidney and a left kidney. Two ureters, the tubes that go from the kidneys down to the urinary bladder, and one urethra. Females about 1.5 inches, males around 6 inches. Now these kidneys are retroperitoneal. This is the peritoneum. I have it boxed right here. So this layer here, it's kind of a little thick layer here. This is the peritoneum, the serous membrane that it lines the organs and the wall of the abdominal cavity. That's why it's called peritoneum. Well, you see the kidneys are behind that. So the kidneys are not covered by the serous membrane, not covered by the peritoneum. Now it says the kidneys, they remove and restore selected amounts of water and solutes. They do. They're going to filter your plasma, and things that you have excess amounts of are going to be voided out. And things that materials like ions and other molecules and water even, that you may need to conserve or reclaim, they're reclaimed from the material that the kidneys are producing. They excrete selected amounts of various materials or waste, like urea and creatinine. Those are waste products. Urea is a way to get rid of excess hydrogen ions. It's NH4. They help regulate blood pressure, okay, by getting rid of certain amounts of water. And you can see here that the right kidney is somewhat, just slightly lower than the left kidney. This is due to the liver, which is on the right side, taking up space on the right side and is superior to this kidney. So it's depressed a little bit. They are protected by the 11th and 12th pairs of ribs. As you can see on this little picture kind of up here, it's your last ribs. So it goes in your blank as 11th and 12th pairs of ribs. These are your vertebral ribs. If you remember, you call them the floating ribs because they go from the vertebrae and float. There is an indention. You have a blank there called the hilus, H-I-L-U-S, or the hilum, H-I-L-U-M. They both mean the same thing. That is the definition there, an indention where blood vessels enter and exit from the kidney. So your renal artery goes in, renal vein comes out. And the ureter, which is going to convey urine from this renal pelvis here that has a star on it. The ureters exit from the renal pelvis, and there's the ureter. And they go down to the kidney, to the urinary bladder. There are 3 layers of tissue that protect and support the kidney. The first is called the renal capsule, and it's R-E-N-A-L. And the second word is capsule, C-A-P-S-U-L-E. And they have fibrous capsule here because it is fibrous. The second thing is an adipose capsule. And see, it has peritoneal fat capsule, but it's an adipose capsule, and all this adipose helps to secure or hold the kidneys to the posterior abdominal wall. People that lose a tremendous amount of weight, like marathon runners, can run into problems where their kidneys will tend to fall, and that can't last that way. They have to go in and have those resuspended. So you have adipose capsule, the fat deposits around the kidney, or kidneys. Around 12% of your body fat is surrounding your kidneys to help support and hold them to the back of your abdominal cavity. And then number 3 is the renal fascia, R-E-N-A-L, Fascia, renal fascia. Looking back at this slide, the renal sinus, S-I-N-U-S, is this place where the blood vessels are, what I'm pointing to right here. It's the space that this renal pelvis sits in. So it's actually the space that the renal pelvis occupies. And I have the definition there, a cavity in the kidney in which the renal pelvis can be found. Now the cortex, C-O-R-T-E-X, I have it marked here, look on the bottom right, renal cortex. The area of the kidney from the base of the renal pyramid, this triangular structure here is called a renal pyramid, R-E-N-A-L, second word, P-Y-R-A-M-I-D, and you have several of them here. There's 1, 2, 3, 4, 5, 6, there's 7 of them you can see on this model here. The medulla, look up here at the top left, renal medulla, the area which contains the renal pyramids. So it's going to be a band of tissue that has these pyramids in it, so it's pretty thick area. So it's the region that contains the pyramids that's called the renal medulla. Now we have renal pyramids. All right, here's the word over here. Triangular shaped structures in the medulla which appear striated. Striated means like little lines. You can see a whole bunch of little lines in here on all the pyramids. Appear striated due to the presence of collecting ducts and blood vessels. So there are a lot of, you can see the collecting ducts in here, which we'll look at right here. You can see those. And a lot of little capillaries are in there too, and it makes it look like it has lines. So it's a striated area. The renal papilla, P-A-P-I-L-L-A, is the tip of the pyramid. So the papilla is the tip of the renal pyramid. This is where, you can see up here at the top, where the collecting ducts are going to drain their urine into the renal pelvis. Now we're going to talk about these little cups that catch that urine in just a minute. Renal columns, a renal column, C-O-L-U-M-N, portions of the cortex between the pyramids. So you have it pointed out right here. This is only this place right here, and over here you can see renal columns, which are in the medulla, but they're portions of the cortex which dip down into the medulla. Now here's a word you have to spell out called parenchyma. P-A-R-E-N-C-H-Y-M-A. Parenchyma, the functional portion of the kidney which contains the nephrons. That's going to be the cortex and renal pyramids area. The renal pelvis. This is where I have my star on this picture. Renal pelvis, a large central urine collecting area. So all these little renal papilla are going to be draining urine out of them from these collecting ducts. It's going to drip out the tip of the papilla, and it's going to be caught by these little structures, which is your next word. Calyces, C-A-L-Y-C-E-S, and singular is a calyx. Cup-like extensions of the renal pelvis. Here's the renal pelvis, and you see that there are little cup-like extensions that wrap around the papilla. So cup-like extensions of the renal pelvis which encompass each of the renal papilla. And when the collecting ducts deposit their urine out the tip of the papilla, these calyces capture it and channel it toward the renal pelvis. You can see that here are some small little cup-like structures that are joined together to make a large one. These are called minor calyces. This is a major. The major delivers the urine to the pelvis. So here's a minor, there's a major there. The nephron. The nephron is the functional unit of the kidney. Nephrons are the functional unit of the kidney. Now I gave you some functions. Functions include filtration, F-I-L-T-R-A-T-I-O-N, reabsorption, R-E-A-B-S-O-R-P-T-I-O-N, and number 3 is secretion, S-E-C-R-E-T-I-O-N. So secretion is what's not reabsorbed. So when the filtrate is made, the material that leaks out of the capillary into the nephron, some of it is reabsorbed and the rest is secreted. So we look at the parts of the nephron. Consists of two portions. Well, the first one is shown here pretty easily. The renal corpuscle, which is what this is, the renal corpuscle. It contains two things we'll look at in just a minute. So number one is renal corpuscle, C-O-R-P-U-S-C-L-E. And number two is the renal tubule, which is a little harder to see on this picture. You can see it over here a little easier. The proximal convoluted tubule, this is a twisted tubule closest to the glomerulus. And there's a loop of the nephron. And the distal convoluted tubule, the convoluted or twisted tubule further away from the glomerulus. That's how they're named. So the renal tubule has three portions. The proximal convoluted tubule, the loop of the nephron, which is loop of Henle, is an old term, and the distal convoluted tubule. I'm going to blow this up where you can see the glomerulus here. So that little blank there is glomerulus. G-L-O-M-E-R-U-L-U-S. A capillary tuft, little capillary ball here. Within, it means inside of, the spherical glomerular capsule. Right here, here's the glomerular capsule. Blood enters this capillary bed by way of the afferent arteriole, A-F-F-E-R-E-N-T, and exits by way of the efferent arteriole, E-F-F-E-R-E-N-T. Afferent means toward, E means away from. Another blank is the glomerular capsule, G-L-O-M-E-R-U-L-A-R. Second word is already written in, capsule. See, it says capsular epithelium here. This is the glomerular capsule. Now look at what it is. It says also called Bowman's capsule. That's an old term. The parietal layer, this is the outside. Remember, parietal means wall, or sac. It means wall around something. The parietal layer is composed of simple squamous epithelium. So all these little cells here that are gray here are simple squamous epithelium. This parietal layer invaginates, it goes in, and becomes the visceral layer of the glomerular capsule because it covers the glomerulus. So it's going to go in and it's going to cover this capillary bed here. Now on this model or this image here, this half on the right is missing that covering. So you can see the capillary bed and you can see the little holes in the capillaries called fenestra. The one on the left side of this image is showing the visceral layer of the glomerular capsule. These are called podocytes. And we'll name those in just a minute. We'll name them again. So these are called podocytes. So the parietal layer of the glomerular capsule is simple squamous epithelium. When it goes in and it covers this entire capillary bed, the cells differentiate into another cell which is called a podocyte. So the visceral layer is podocytes. Let's go back over here and look at something. This is your nephron here, right? The corpuscle and the tubular portion. And here is the collecting duct. So there are many nephrons that are going to join these collecting ducts. Here's another picture. There's another model, and you can see that there are several. We have two nephrons here, but you have several extensions here where other nephrons are joining to this collecting duct. So we have two here. One's over here, and one's over here. So collecting duct. A duct to which many distal convoluted tubules join and deposit their urine secretion. Collecting is C-O-L-L-E-C-T-I-N-G. Collecting is collecting duct. Now here's the pyramid here. This is the renal pyramid, this triangular shaped structure here. And if that's the pyramid, the tip down here is the renal papilla. And so this part of the collecting duct is called the papillary duct because it's down in the renal papilla. So P-A-P-I-L-L-A-R-Y, duct, the end of the collecting duct as it terminates at the end of the renal papilla. Nephrons can be named according to their positioning in the cortex. Your first blank here where it says blank nephron, that's a cortical nephron. That's going to be the one on the left. See, they labeled it down here. C-O-R-T-I-C-A-L, cortical nephron. Has this glomerulus, there's the glomerulus there, in the outer portion of the cortex, toward the outside, to the top part of the cortex. And its loop of the nephron penetrates the outer portion of the medulla. So see, it barely goes in the outer portion of the medulla. Doesn't go very far down in there. That's a cortical nephron. Well, let's look at the next one. It's called a juxtamedullary. It means in the vicinity of the medulla. Juxtamedullary, nephron. Has this glomerulus deep in the cortex. So see, it's way down close to the medulla. There it is, deep in the cortex. And this loop of the nephron penetrates the medulla almost to the renal papilla. So see, the loop of the nephron goes pretty deep into the medulla. So those are the two types of nephrons, cortical nephrons and juxtamedullary nephrons. And we have histology of the nephron, and we're talking about this endothelial capsular membrane. This is the area where filtration in the nephron occurs. It's going to be across this capillary bed, and the materials that leak out are going to be called filtrate, and the filtrate is going to go into this capsular space, which is lightly violet here, and then leave by way of the proximal convoluted tubule. So let's look at this endothelial capsular membrane here. Your first word is endothelium. Endothelium, E-N-D-O-T-H-E-L-I-U-M. Remember, endothelial lining is continuous throughout the entire cardiovascular system. The heart, arteries, and veins have a simple squamous epithelial lining, and it's called the endothelial lining. In the heart, it's called the endocardium. So let's read the definition. The simple squamous epithelium, which comprises the glomerular capillary tuft, which makes up that capillary bed there. This capillary has fenestra or pores. Fenestra is Latin for window, or it can be pore. And it says they're 0.5 to 1 microns in diameter. So look at all these little holes here, these little fenestra. 0.5 to 1 micron. Well, what that stops from leaving the capillary are cells, and even platelets don't leave. Cells, remember the red blood cell is between 7.5 and 7.7 microns. Huge, and red blood and white blood cells are larger than that, so there's no way they're getting through that first line of filtration here, that first barrier. These pores are too small for blood cells to pass through. True. Now what they don't show on here, but they do mention in this writing on the right, is the basement membrane. It says it's been removed. An extracellular fibrous glycoprotein matrix, which acts as a dialyzing membrane. Remember, dialysis membranes have molecular-sized holes in them, like you would use in lab the dialysis membrane for doing osmosis and diffusion experiments. It functions to screen out large molecules from leaving the plasma. So the basement membrane, the glycoprotein basement membrane here, is a dialyzing membrane, and it selects by the size of the molecule, so it won't let large molecules like proteins go through here. So proteins are blocked from leaving the plasma. At least large proteins are. And then here's the visceral layer of the glomerular capsule. Remember, these are called podocytes. They're part of the visceral layer. And these podocytes, there's a nucleus there and a nucleus for this one here, have extensions called pedicels. This means little feet. So they form, the filtration slits formed by the podocytes, P-O-D-O-C-Y-T-E-S, of the visceral layer of the glomerular capsule. Extension from the podocytes are called pedicels, P-E-D-I-C-E-L-S. It means little feet. And they form, when they interlock, they form what's called filtration slits. Well, there happens to be a thin membrane called the slit membrane, which extends across the filtration slits and restricts the passage of medium-sized molecules. So there's another dialysizing membrane that's in these filtration slits that won't let medium-sized molecules get out. So what type of molecules are going to get out to form the filtrate inside of this glomerular space here? That's right, small ones like water, glucose, sodium, potassium, calcium, hydrogen ions, sodium chlorine, that kind of stuff. So let's look at the blood and nerve supply. We're going to look at the blood supply, the circulatory pathway for blood going through the kidneys. So we have blood circulation to the kidneys is under motor control, which regulates the diameter of capillaries. The renal arteries, this number 1 here, I have everything numbered, transports 25% of the total cardiac output to the kidneys per minute. That's around 1,200 mils. You've got between 4 and 5 liters. So that means that every 4 minutes, your entire blood volume should be cleansed through your kidneys. It says to be cleansed, yeah. Salts, glucose, urea, other solutes are going to be filtered out of your plasma and determined whether you need to reabsorb them or secrete them in the form of urine. So the renal arteries are going to bring the blood to the kidneys. So in your reading here, the renal arteries branch into segmental arteries, S-E-G-M-E-N-T-A-L arteries. These little branches off of the renal artery are called segmental arteries, which in turn branch into interlobar arteries. Interlobar arteries go between the pyramids. So you can see these branches go between the pyramids. So I'm going to get a better picture. See, there's interlobar vein. Interlobar artery goes in number 3, is going in between the pyramids. So let's look over on another slide. Here come the interlobar. See, I have it labeled again. Number 3, arteries on the side of the pyramids, which branch, and they're going to go along the base of the pyramid as the arcuate artery. A-R-C-U-A-T-E artery, which branch to form, and scratch this out in your notes because there's a new term, the cortical radiate artery. C-O-R-T-I-C-A-L, second word, R-A-D-I-A-T-E, and third word is just artery. You see how a radius goes from the center out to the periphery of a circle or something? It's going from the center out. So they're called cortical because they're in the cortex and they're radiating out away from the arcuate toward the capsule, and they're arteries. Now, branching off of these cortical radiate arteries, look over and let's look at number 6. Yeah. You have a branch coming out and going to form the glomerulus inside of this glomerular capsule. Now, the one on the right is where I have a number 6. Look on the left, you can see the glomerulus, number 7. That's the glomerulus. So you can also see an afferent coming off of the cortical radiate, going to form the glomerulus, and then coming out, look on the far left here, the one coming out is called the efferent arteriole. So the efferent arteriole now leaves the glomerulus and branches to form a capillary network right over here. Here's one of them called the peritubular capillaries. P-E-R-I-T-U-B-U-L-A-R capillaries, which surround the convoluted tubules in the cortex. Now in this model, they took these capillaries and put them off to the side so you can see the proximal convoluted tubule, the loop of the nephron, and the distal. So you can see those a lot easier if they're moved off to the side. But the peritubular capillaries, they're going to be around the proximal and distal convoluted tubules. Well, there's a second capillary bed that's also formed just down here, also forms the vasorecta, number 9 there, the vasorecta, V-A-S-A, second word, R-E-C-T-A, which surrounds the loops of the nephron. So these would be surrounding the loops of the nephron on both of these nephrons here. But again, they're put off to the side so you can see everything nice and clean. They both unite to form the cortical radiate vein. So let me see, put it, it's this blue one right here. Cortical radiate veins right over here. I didn't put that on there. That's the cortical radiate vein on the far left. Oh, it's number 10. That's what it is. Number 10 is the cortical radiate vein. C-O-R-T-I-C-A-L. Second word, R-A-D-I-A-T-E, and then vein. Those become the arcuate veins, number 11. Then the interlobar vein, there it is right there, number 12. Then they form together to form segmental veins. Here's a segmental vein here. I only show really one good one there. It's segmental veins. And then finally, all the segmentals join together to form the renal vein. And that goes back to the inferior vena cava, which is going to be circulated through the heart and back out to the body. I'm not going to spend very much time on this next one, the juxtaglomerular. There's just a lot to this one. The juxtaglomerular apparatus, which you can see here at the bottom, it's got a bracket there, is formed by the distal convoluted tubule, okay, and the afferent arteriole. So these two right here. And their job pretty much is to regulate blood pressure and the filtration rate of the glomerulus. Now it talks about something called the macula densa. It has a similar effect. The macula densa cells here is part of the distal convoluted tubule. It's going to have a similar effect to regulate blood pressure and the filtration rate of the glomerulus. The juxtamedullary cells are called in the mesanglial cells here. It's all these cells here, these little gray guys here. Now they secrete renin. So the juxtaglomerular cells secrete renin. That stimulates the release of angiotensin, which stimulates the release of aldosterone from the adrenal cortex. So I have in your notes here, release renin. Its release causes the formation of angiotensin, which constricts the afferent arteriole, thereby controlling blood flow to the nephron. Okay, so it also is going to have an effect on your blood flow going through the glomerulus. So again, I'm not going to dwell too much on that right now. Moving on to the physiology of the nephron, we have some things to talk about before we talk about this, this slide. 3 functions. Once again, controls blood concentration and volume by removing selected amounts of water and solutes. Helps to regulate blood pH. Removes toxic cellular waste products from the blood. Urine formation requires 3 principal processes: glomerular filtration, tubular reabsorption, and tubular secretion. So filtering of the plasma, reabsorption of some of it, and secretion of the rest of it. So you have a blank there, and what goes in the blank is filtration. F-I-L-T-R-A-T-I-O-N. Occurs by forcing the fluids and dissolved substances through a membrane by an outside pressure. It occurs across this endothelial capsular membrane that we looked at a minute ago, which is right here. The pressure is blood pressure. That's the driving force there. The fluid that's going to leak out into the yellow space here, the capsular space, is called filtrate. Now, adaptation of the renal corpuscle for filtering blood. A capsule of highly coiled glomerular capillaries represents a vast surface area for filtration. So this capillary bed here, the glomerulus. The endothelial capsular membrane that we looked at a minute ago, right here, is adapted for filtration. Fenestra, basement membrane, slit membrane. The efferent arteriole, the one that leaves, is smaller, smaller, S-M-A-L-L-E-R, in diameter than the afferent. So since you have a large vessel coming in and a small vessel going out, it builds up pressure in this glomerulus. You can experience that if you turn on the water hose full blast. You'll see that it might shoot that water out like 6 feet. But if you put a restriction on there like a brass nozzle, it might shoot it out 15 or 20 feet. A lot more pressure in that hose and the glomerulus because the exit vessel is smaller. The glomerular capillaries have the highest blood pressure of all capillaries, the 40 to 60 millimeters of mercury there. The endothelial capsular membrane is very thin. So when we look at this, this membrane here that's formed, it's very thin. It says 0.1 micrometer and separates blood cells and large proteins from the glomerular capsule. So blood cells and large proteins can't get out into this capsular space here because of the fenestra and the basement membrane. All right, so let's look at something called net filtration pressure. It's also called effective filtration pressure. And let's look at the definition. The pressure which causes filtrate to be formed, that means to leave the capillary and enter the glomerular space. The space here where it's yellow. Now you have an equation. I don't have it on here, but you have it in your notes. It's net filtration pressure equals, that'll be glomerular hydrostatic pressure, could be glomerular blood hydrostatic pressure, minus two resistance forces, the capsular hydrostatic pressure and the blood colloidal osmotic pressure. So let's look at your definitions. The first one is capsular hydrostatic pressure. C-A-P-S-U-L-A-R. Second word is hydrostatic. H-Y-D-R-O-S-T-A-T-I-C. Your definition says it's going to be this green arrow here. The force which a fluid under pressure exerts on the wall of a container, the capsular wall. And so you see this resisting being filled. This glomerular capsule is going to resist being filled just like a balloon would. When you fill a balloon with air, it's hard to push that air in there, and it wants to recoil and get that air out, right? This is the recoil pressure of this capsule, the resistance to being filled. The second one, blood osmotic pressure. Blood, B-L-O-O-D. Second word, osmotic, O-S-M-O-T-I-C. So let's look at this definition. The pressure which develops from water movement into a contained solution. It always develops in the solution with the higher concentration of solutes. Since blood has more proteins than filtrate, water moves out of the filtrate and back into the blood vessel. All right, that's going to be these little, I guess, red lines here. You see the water's trying to move back in. When the filtrate's formed, not only are solutes leaving, but a lot of water is leaving. And when a lot of water leaves, there's more protein inside of the capillary bed and less water. And so water is going to try to move osmotically back in. And that's the blood osmotic pressure. Your third blank is the glomerular hydrostatic pressure. G-L-O-M-E-R-U-L-A-R. Second word, hydrostatic, H-Y-D-R-O-S-T-A-T-I-C. The blood pressure in the glomerulus, that's the driving force, this purple arrow. This pressure is pushing against the walls of the capsule and the filtrate that's already there. So look at your millimeters of mercury represented for each of these forces. In your equation, you can write this down. Net filtration pressure equals the glomerular blood hydrostatic pressure, 55 millimeters of mercury, minus capsular hydrostatic pressure, 15 millimeters of mercury, plus 30 millimeters of mercury for blood osmotic pressure. So that's going to be 50 minus, 55, I'm sorry, minus 45. So the net filtration pressure is a positive number, 10 millimeters of mercury. And as long as it's a positive number, like it is right here, there it is, filtrate can be formed. As long as the driving force, your blood pressure, is greater than the two resistance forces, capsular hydrostatic pressure and the blood colloidal osmotic pressure, water trying to enter back in the glomerulus, as long as that's positive, there is a formation of filtrate. It says net outward pressure, so it's going to go into the proximal convoluted tubule. Tubular reabsorption, the movement of the filtrate back into the blood of the peritubular capillaries or the vasa recta. So the capillaries surrounding these tubules here, the proximal and distal convoluted tubule, or the loop of the nephron. Now something you need to look at is look at the cuboidal cells that make up the proximal convoluted tubule. Here it is, proximal convoluted tubule cells. They have a whole lot of microvilli. Microvilli are finger-like folds of the plasma membrane, and they increase the surface area for reabsorption of materials from that filtrate. When you get down to the distal convoluted tubule, you see that there's not a whole lot of microvilli, not a whole lot, so you're going to get less reabsorption there. About 90% of what's going to be reabsorbed is going to be reabsorbed immediately by the proximal convoluted tubule. Okay, so whatever comes out in the formation of the filtrate in the glomerular space, when it leaves that glomerulus and it goes into the proximal convoluted tubule, about 90% of that is going to be reabsorbed immediately back into the peritubular capillaries. And I gave you some things that will leak out: water, glucose, amino acids, sodium, potassium, calcium, chlorine, bicarbonate, urea. A lot of stuff leaks out, and a lot of it's also reabsorbed. The tubular max, the maximum amount that can be reabsorbed. The rest is voided. This allows the body to retain most of its nutrients. Okay, can retain most of its nutrients. The kidneys and these nephrons are built in such a way that they monitor the levels of these materials in your bloodstream, and what you have excess amounts of will not be reabsorbed, like water will not be reabsorbed, and certain electrolytes will not be. A blank. Urea, U-R-E-A, is only partially reabsorbed. Okay, so you got to have some in your plasma, just like you have to have some carbon dioxide in there. The carbon dioxide is important to maintain your pH. This urea is also important to help maintain pH. Another blank is glucose, G-L-U-C-O-S-E, glucose. Reabsorption probably involves a carrier system, some kind of a carrier protein. Sodium ions are reabsorbed actively. That means it requires the energy of ATP to reabsorb the sodium ions from the proximal convoluted tubules back into the peritubular capillaries. And we saw earlier up here how the proximal convoluted tubules contain microvilli, M-I-C-R-O-V-I-L-L-I, which increase the surface area approximately 20%, or 20 times, I'm sorry, 20 times. The sodium ions are actively transported through the cells and into the peritubular capillaries. So they're going to be actively transported from the filtrate into the cells of the peritubular capillaries. Now, sodium ion concentration inside the cell is higher than in the lumen inside the tubule. It's higher in the cells around the tubule. The blood is now slightly more electropositive because it's going to be the peritubular capillaries are there too. And chloride ions follow sodium ions out of the tubule by electrostatic attraction. So chloride ions are also going to leave the filtrate because they're negative ions. They're going to follow the positive ions, which are sodium, into the cells and back into the bloodstream of the peritubular capillaries. Because you have a whole lot of electrolytes that are being reabsorbed, water, it goes in that blank, water, follows since the proximal convoluted tubules are always permeable to it, and the cells have a lot of electrolytes in the cells compared to what's in the filtrate. So the filtrate has more water and less solutes. So it's going to go towards more water towards less water. And so water is reabsorbed by osmosis. Due to the sodium being transported back into the blood, the osmotic pressure of the blood becomes higher than the filtrate. Water follows the sodium ions into the blood to reestablish osmotic equilibrium. This is called obligatory reabsorption. OBLIGATORY, reabsorption. When the blood water concentration is too low, ADH is released. Remember, antidiuretic hormone is going to make your kidneys retain water. ADH makes the membranes of the distal convoluted tubules and collecting ducts more permeable to water reabsorption by a carrier molecule. This is called facultative reabsorption. FACULTATIVE. That means it's assisted or helped, facilitated. Now, still we're on volume here. That should be 1,000 to 1,800 milliliters eliminated per day. 1,000 to 1,800 mils eliminated per day. That's going to depend on your diet, basically. So it says volume is influenced by, and I've given you several things here. Blood pressure is high. That means you have too many fluids in your bloodstream, and you're going to get rid of excess fluids. The blood concentration kind of goes along with that. Your diet, whether you're eating dry things or materials that have a lot of water in them. Temperature, if you're hot, you're going to be sweating. You're going to be losing water through sweat. And so you'll have less water in your blood. Diuretics, remember diuretics are materials like caffeine, cokes, coffee that will make you lose water. So they cause the kidney to lose water. The nephrons will not reabsorb it. The mental state, if you're anxious, you tend to urinate more than if you're depressed. Your general health will have an effect on the volume. Now, tubular secretion, SECRETION. This process adds materials from the blood to the filtrate. And I just listed some things here, and you see potassium, hydrogen, ammonium ions, urea, creatinine, penicillin, stuff like that. And you know that happens because if you're on antibiotics, you know what antibiotics smell like when you have to take them, right? There's a smell to the antibiotic tablets, those little capsules. And when you go urinate, it smells just like that because your kidneys are going to filter out the penicillin. That's why you have to keep taking it on so many hours to keep it up in your bloodstream because your kidneys are constantly filtering that out. This process functions to rid the body of certain materials, excess materials, water and electrolytes and stuff, as well as help control the blood pH. Many foods are acidic, like drinks like Cokes, coffees, teas, fruit juices, salad dressings, like vinegar on your salads. And the body tries to maintain a pH of 7.35 to 7.45. That's because that's where your enzymes work. That's the optimum range for enzyme activity in your body. You get above or below that, and you're either going to be acidotic or alkalotic, and the enzymes start to denature and become non-functional, and you're in physiological stress and danger. To raise the blood pH, the renal tubules secrete hydrogen ions and ammonium ions into the filtrate. Normal urine's a pH of around 6. It tends to be around 6 to 5 because of all the acidic products that we ingest. The countercurrent multiplier mechanism has to do with the convoluted tubules, a process which allows the kidneys to secrete hypertonic urine or hypotonic urine. If you have plenty of water, your urine is more watery, more voluminous, and if you have been sweating a lot or you don't have enough water, it becomes more concentrated. So this can control the amount of water that makes up your urine. Now another blank, the ureters, URETERS, deliver urine from the kidneys to the urinary bladder. Here's one on the right and one on the left side. They're approximately 10 to 12 inches long, and you have a blank, peristalsis, Peristalsis. That's wave-like muscular contractions. We'll move it from the down the ureter toward the urinary bladder. Hydrostatic pressure, that's, more and more urine is being formed continuously because of your blood pressure, and so more filtrate's being produced in by your glomeruli into the capsular space. And gravity, Gravity, water runs downhill, helps move the urine. There are three coats. The mucosa, remember the mucosa is the endothelial lining here, which is going to be transitional epithelium, which is going to line the inside of the urinary bladder and the ureters, and the inside lining of the ureters also. The muscularis, which is the muscle, which we'll talk a little bit more about in just a minute, is smooth muscle. And fibrous on the outside, going to help hold the urinary bladder and the tubules in place, as well as the peritoneum down there in the pelvic cavity. So we look at the urinary bladder in general, a hollow muscular organ, and it is hollow, you can tell there, held in place by the peritoneum. And there's peritoneum pointing to the surface. It also fans out and secures the ureters a little bit and holds the urinary bladder in place. So look at your next little blank here is trigone, Trigone, a smooth mucosal layer. Okay, it's going to be a smooth muscle because the bladder is made of smooth muscle. The ureters drain into the urinary bladder at the base corners. See up here at the top? This is where the ureters actually enter the urinary bladder. They're called uteric orifices, openings of the ureters. And so urine comes from these ureters and it drains into the urinary bladder. There are four coats of the urinary bladder, so we're going to go back to one of them. Number one is mucosal, M-U-C-O-S-A-L, the inside epithelial lining, composed of transitional epithelium. The inside lining contains folds called rugae, R-U-G-A-E, which allow the urinary bladder to stretch as it fills with urine. It's going to fill. The submucosa, which you really can't see on here, connective tissue layer which holds the mucosal layer to the underlying muscular coat. So it's going to be like the lamina propria. It's called the submucosal layer, holds the epithelium to the muscle. So number three is muscularis, or detrusor muscle. And there it is right here, detrusor, which is basically just pointing to the muscle. And it has several layers here we're going to talk about. 3 layers of muscle, inner longitudinal, middle circular, and outer longitudinal, which they don't show on this picture. But they do show the sphincters. The internal sphincter, it's called the internal urethral sphincter, I-N-T-E-R-N-A-L. You can barely see it here. It's a little darker pink here. It's right where the urethra leaves the urinary bladder. There's a sphincter there, a circular muscle called a sphincter, made of smooth muscle, and its name is the internal urethral sphincter. Well, as that urethra goes through the floor of the pelvis, which is called the urogenital diaphragm, it's muscle there. As it goes through, there's another sphincter that surrounds the urethra, and it's called the external urethral sphincter, E-X-T-E-R-N-A-L, and this one is skeletal muscle. So the internal is smooth muscle, the external is skeletal muscle. Don't worry about adventitia, that's going to be the peritoneum. So we have another word for the expulsion of urine. Expulsion of urine. This is called micturition. M-I-C-T-U-R-I-T-I-O-N. Expulsion of urine from the urinary bladder. We call it urination, or when you go TT, it's voiding, getting rid of the urine. The average urinary bladder capacity is between 400 and 800 mils. A can of Coke is roughly a little more than 400 mils. A bottle of water is 500 mils. So you can see approximately how big that urinary bladder is going to be. Now the next blank is urethra. U-R-E-T-H-R-A. A tube extending from the urinary bladder to the external urethral orifice, the opening there where urine comes out, where urine is expelled. And your last little blank there is uremia. U-R-E-M-I-A. Toxic levels of urea in the blood due to the kidneys not functioning correctly. So they're not getting rid of the excess amounts of electrolytes and other molecules, and they're raising to toxic levels in your bloodstream. That's what happens to people who have to be on dialysis. If they don't get on dialysis, waste materials build up and they enter into that state, which is just uremia. Now, I put one more slide on here. This is an electron micrograph. It's like a scanning electron micrograph. And you can see these glomeruli, little capillary balls here, and you can see the little blood vessels, probably afferent and efferent. Afferent going out, yeah. Coming in, efferent going out. So you can see that on this picture. I just put that for your viewing pleasure. And that's the end of this lecture.